Dober večer, prijatelji znanosti, prijatelji Univerze v Novi Gorici. Pozdravljeni na naslednjem v vrsti znanstvenih večerov, ki jih organiziramo tukaj v dvorcu Lantieri. Nocojšnji gost in predavatelj je profesor Francisco Baralle, po rodu Argentinec, dolga leta pa dela v bližini v Trstu. Dovolite, da ga nakratko predstavim. Professor Baralle obtained his PhD from University of Buenos Aires in chemistry, and his second degree is in medicine and surgery obtained from University of Naples. Soon after graduation in 1969, Professor Baralle transferred to the Institute of Biochemical Research, Foundation Campo Mar, which was directed by Professor Luis Leloir, the recipient of Nobel Prize for Chemistry in 1970. 4 years later, in 1974, he moved to the Medical Research Center, Laboratory of Molecular Biology in Cambridge, where he worked under direction of another Nobel laureate, Dr. Sanger, who received the Nobel Prize in Chemistry twice, one of the 4 people who ever received Nobel Prize twice in their life. From 1980 till 1990, Professor Baralle was a lecturer of pathology in Cambridge at the Magdalen College, and after this period, he was appointed the director of the Trieste branch of the International Center of Genetic Engineering and Biotechnology. In 2004, he became Director General of the same institute. For his research achievements, Professor Baralle was given several awards and became member of important institutions such as European Molecular Biology Organization. In 2001, he became the member of the Argentinian Academy of Science. In Argentina, he also received the Platinum Conex Prize for Science and Technology, which is given to the scientists of the decade in genetics and cytology. And last but not the least, Professor Baralle is also recipient of the Golden Plate Award of the University of Nova Gorica for his contributions to the development as well as affirmation of our university. Let's now move slightly towards the topic of today's presentation, which is of course based on achievements of Professor Baralle in science. And if I make my choice, let's say, in 1977, Professor Baralle made one of his most important contributions by publishing the first complete primary structure of an eukaryotic messenger ribonucleic acid. This is the messenger RNA coding for beta globin, which to most of us is known as a hemoglobin. Two years later, his group isolated the gene for epsilon globin, which is a component of the human embryonic hemoglobin. In the 1980s, Professor Baralle was one of the first to describe the pre-messenger RNA alternative splicing process, and since then he has made crucial contributions to understanding the molecular mechanisms involved in these cellular processes important in health and disease. And I believe we'll hear more about this. Therefore, Professor Baralle, the floor is yours.
Okay. Well, thank you very much. And I am later if you want. I am really glad to be here again. I am very fond of University of Nova Gorica, although at times I have difficulty in finding it, like tonight, that I couldn't find the building. But the university has been great also for us because we have a very good relationship, and many of our PhD students are registered here, although they do the research in ICGEB Trieste. So we keep thinking that we should expand this, even in moment of crisis, and every year we send a few. This year we have some problem with the recruitment of PhDs, but we'll pick up again next year. So I was told that I should be sort of for a general audience, so I will essentially start from the beginning, and then towards the end of the talk, I will do some about my subject, that is pre-RNA splicing, that is rather boring and sort of arid, very technical, but that has led us to some research in neurodegeneration that I think is very exciting and very near application. So let's start, as I said, from the beginning, and most of you would know, but if not, I'll tell you now. The basic of all our living organisms is a cell. And the cell has a nucleus, and within that nucleus, there are chromosomes. The chromosomes pack inside them very long, over 2 meters or more, DNA molecules. And these DNA molecules have a sequence, a structure that code for all the functions of the cell. The structure of the DNA was elucidated by Watson and Crick in Cambridge, actually in the LMB, where I went to work about 20 years later after they have discovered this. But you know, structure is a structure, and although shapes are nice, and the fact that you have base pairs and helixes and so on, did not tell us a lot about how the cell functions. So a nice structure should be or for living or for driving, but we should know how it works. And the merit to knowing how it works and how it's really made goes to Frederick Sanger. As was mentioned before, he was Nobel Prize for Chemistry in 1958 and 1980, and I want to take advantage to make a small speech about him because unfortunately he passed away 2 days ago at the age of 95. He is such an extraordinary person that at 65, 3 years after winning his second Nobel Prize, he stopped working and went into retirement to garden and to build a boat, a sailing boat all in wood, all by himself. Never went back to science. Obviously, difficult to do anything more than that, I suppose. So he did well after 65 to retire. But at the beginning, in the 50s, he elucidated the structure of the first protein, insulin, and the amino acid sequence. And in the 70s, actually I have the big luck to be there at the time, he developed the DNA sequence methodology that it has made a tremendous jump, and most of you know. I mean, this was what we were doing. This it was called a bidimensional homochromatography at the time, that I did myself in 76, 77. And at that time, this method that was called the wandering spot method would allow you to sequence 10 nucleotides of any sequence in a week. Because you have to prepare this bit of RNA, digest it partially, and then run in 2 dimensions, one by charge and one by size. And then according to the mobility, you could read which nucleotide was lost from the chain. So if the chain was GAA, GAT, GAA, this is all the nucleotides of the DNA or RNA. A small jump to the right was a T. A big jump vertical was an A. A small jump to the left was a C. And that was the way that we were building up sequences. After Sanger developed the method that is called dideoxy in the 80s, we could do 100, 2,000 nucleotides a day with the new machines that we have in the labs today. Although there are super machines like this one that can do more or less 3 × 10⁹ nucleotides, so the equivalent of a human genome in one day today. So only 30 years pass. And the improvement from here to this is amazing. That has resulted in the publication of the human genome. This was in 2001, and was done and completed in what more rightly was called the Sanger Institute in Cambridge, and also in the NIH in the States, but there was more also another private company that did that as well. Now, let's go back to the DNA. The DNA is a double-stranded molecule with base pairs of guanosine and cytosine, or adenosine, thymidine, and that wraps up around and replicates, copying itself. Now, the important thing is that this DNA have eventually to code for proteins. And these proteins are coded by the genetic code. That is, every 3 nucleotides mean 1 amino acid. GCA means alanine, AGA means arginine, for example, in this case. Some codons are degenerated, so there are 3 or 4 for each amino acid. GAT is aspartic, and so on. So, and the incredible thing that always amazes me is that it's universal. The code in a bacteria, in a fungi, in a lion, or in us is always the same. Always, always, GCA will be alanine, and AGA will be arginine. And that allows us to cross species once we learn how to mobilize DNA. So we could clone a piece of human DNA, put it in a bacteria, and make it express the human protein. That's the basis of the biotech industry, essentially. At times, the DNA gets damaged because ultraviolet light, radiation, chemicals, and so on. And this damage from a code that makes glutamic acid, for example, GAA, this is the DNA copied to the messenger RNA, translated to a protein. If this adenosine is mutated and is mistakenly copied as uracil, this will be GUA and codes for valine. And a little change causes a big disaster in the organism. This is the mutation of the first molecular disease that was described, and that is sickle cell anemia. Just 1 amino acid change in the hemoglobin protein. So, in summary, just to get to a little bit more in detail. The DNA is a double-stranded molecule that gets replicated, copying itself. It's transcribed by an enzyme called RNA polymerase to RNA that have all the codons in a line and code for different amino acids that make a protein with a defined sequence. Now, it's not that easy. I mean, this is the scheme. A little bit more in detail, you have transcription, you have RNA processing because the genes are interrupted in higher organisms. We'll go to that in a minute. RNA processing, the export from the nucleus to the cytoplasm, the mRNA has to be transported and localized properly in the cell, have to find the ribosome that we translated into protein, and the proteins, if they are folded correctly, will be used. If they are incorrectly folded, will be inactivated and degraded. The mRNA also is controlled, and at times if there is a mistake in the process, and it's an RNA that will make a wrong protein, is recognized and degraded as well. As I told you, in bacteria, the information, the genetic information is contiguous. So the sequence of the DNA, of the RNA, and of the protein is always the same. In humans, in eukaryotic cells, in yeast, instead, if you read insulin, In the gene will be insulin in the protein. In the gene, you don't read insulin. The sequence coding for the protein is interrupted by sequences that apparently are meaningless. And to make the proper protein, you have to process. You remember this step. The process involves splicing out this X, let's say, and joining the in, su, and lin. That is the splicing process. Essentially, the bit of DNA that codes for proteins is called exons, and the bit of DNA that is in the middle is called introns. So exons and introns. Genes come in all shapes, are more or less like the dogs. I mean, you can have little ones with 3 exons and 2 introns, huge ones, 200. There are also genes like the gene, the dystrophin that is involved in muscular dystrophy that have 1 million base pairs. Alpha-tropomyosin, and many of them have several splicing, alternative splicings. So that can be from a single gene, you can make this protein, including this exon, or you can make a different protein, including these ones, and so on. And these versions or isoforms of the protein are specific of a given tissue. So for muscle, you have this one. For a smooth muscle, this other one. For the fibroblasts, for the brain, you have a different tropomyosin. Most of the genes, 80–90% of the genes, have this process. From one gene, 10 proteins, 15 proteins, 5 proteins, and that's what makes the coverage of all the variety that we have in the cell from only 20,000 genes. That is more or less what apparently is in our chromosomes, up to now at least. So let's go a little bit on the splicing. I forgot to, no, I didn't forget to check the clock. The splicing is this process, so the exons will be recognized by different elements, the introns as well, and eventually these exons will be joined together. These U molecules are complexes of proteins and RNA. These are other type of proteins, HRMPs are called, SR proteins. These ones tend to be positive, these ones tend to be negative for the inclusion of this exon. We'll see that in a minute. All this is not done by anything. It's done by a big machinery in the cell called the Spliceosome. And here you have a photo of the Spliceosome. It's a complex of more than 100, 150 proteins and 6, 7 ribonucleoproteins, so complexes of proteins and RNA. The splicing process is very complex. It's not just the linear recognition, and has to be very precise. Imagine that you are the RNA polymerase, the enzyme that has to copy the dystrophin gene that morning. So the foreman that is distributing the work in the cell says, well, today for you is dystrophin. Oh, bad luck. All day work in transcribing it. And you start to transcribe. And behind you comes this guy processing the introns. Imagine if you are in the 900,000 base and this guy makes a mistake and you have to throw away all the work of the day. So this machinery is very, very precise. So, Pol2 RNA polymerase doesn't get angry. It's precise, although it has very little to go for. When the sequence of the RNA is transcribed, the borders of the exon are marked by this sequence, AG or GU. AG or GT, this one, small one here. GU, AG. 3' splice site, 5' splice site. because the RNA is read. If you come reading from here, 5 prime, 3 prime, 5 prime, 3 prime. With this, it's difficult to recognize anything in 200,000 of these bases. So the recognition is made by a series of proteins that will see these areas, but we'll see them because they will be marked by other identifiers that are called exonic splicing enhancers, at times silencers to diminish the recognition, or exonic or intronic enhancers or silencers. And all these interactions lead to a very complex process that is decided like a football match, let's say, no? I mean, you have positive players and negative players. If the positive prevail, you win and get included. If the negative prevail, you get skipped. Okay? So that exon is not included in the protein. Now, I wanted to do a big chat about this, but we will not have time. So just to give you a flavor. If you are, something that I have done for a while, diagnostic genetic disease, inherited disease. Now you can say, well, in a day I sequence the whole genome of the patient and see, well, where there are changes, those changes are the cause of the disease. However, some of those changes don't seem to do anything. They don't change the amino acid. They are in the middle of sequences that don't mean anything. And so many of those are simple polymorphisms, a difference between you and me, like I don't have hair and you do, for example. But at times, those variations could mean that some of these elements for splicing has been disrupted. And then the exon will not be included. So it will be a very serious defect of the protein. Not only will have one amino acid change, but it will have all this sequence missing, that may be 50, 80, 90 amino acids, and will be completely useless. To give you just one example, because we don't have time for more, cystic fibrosis is a disease that can present in a very serious, acute form, or in one that is just only some of the, there is no lung involvement, maybe just male sterility because obstruction of the vas deferens, maybe some bronchiectasia, but not other problems in the lung, or maybe very serious, like normally is, you have problems in the airways, problems in the digestion, and so on and so forth. This depends many times of small variations in inclusion of exons. This is the exon 9 of the gene of the CFTR, 8, 9, 10. Is needed to be included to be a good thing. For some joke of evolution, all of us are very variable and have 90% of this and 10% of this. And that normally is no problem. If you get to 50, 40% of that, then you start to have some of the non-classical cystic fibrosis. And if you get below the 10%, you have the full-blown disease. One reason why it cannot be included at times is this sequence that is just at the end of the intron. TG, 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 T, 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 T. Normally, we need many T's here. And then this is the site of recognition of the exon, where it's cleaved and joined together to the rest of the protein coding sequence. When these T's are 9, it's fine. When these T's are very few, 3, and this TG is very long, practically there is no inclusion. It's all like this, and the individual patient have cystic fibrosis. Now, we invested many years on that and discovered that the problem was that in this UG repeated sequence that is very variable between us, there was a protein that is binding. And this protein recruits others that inhibit the recognition of the exon. It shouldn't be there. It's just there because a joke of evolution, as I say, there has been between rodents and primates a hit of something that is called a transposon, a mobile element that has disrupted the genome and brought with it all these repeated sequences. In that sense, all these elements that help the exon to get included are superfluous if you don't have this. You can see that at times, variations here could change the amino acid and jump the inclusion. I show you one example. No? This is the sequence of the exon 9, and these boxes are elements that help the exon to be included. One is this one, in this area. If there is a mutation that is found in patients between C and A, there is a change of amino acid, alanine to glutamic. This change, though, doesn't affect the function of the protein, that is a chloride channel. But actually, the protein never exists because that mutation disrupts this regulatory element and the exon is totally skipped. This is the band for exon inclusion. This more or less the normal situation. This patient, this mutation instead, as you can see, is all excluded. And this is the cause of the disease. Not the change of amino acid, but the exclusion of the exon. This has also some evolutionary implications, that you cannot change the protein as you please and then select the best one, because maybe at that point you are touching a splicing element and the whole thing will be skipped. But that's a more complex story that I don't have time to tell you. Well, I told you about this protein, and I use this example on purpose, because this protein, we were working on it about 10, 12 years ago. And then 6 years ago, this protein was found to be the main component of the inclusions in the brain of patients with amyotrophic lateral sclerosis, something that's called the Lou Gehrig disease as well, that many sportsmen get, and of patients with frontotemporal lobar degeneration, another neurodegenerative disease. The protein is a fantastic protein. It's an RNA binding protein. It's an HNRMP, it's called. Interacts with this GU nucleotide. That is what it binds, and binds through this interaction with phenylalanine. And the structure and biological properties, so specificity, will always recognize GU repeats in Drosophila. The Drosophila protein or the human protein will do the same. And you can exchange them. I can put the Drosophila protein in human cells, it works fine, and vice versa, as you are going to see. So in this disease, the protein that normally is in the nucleus and shuttles from the nucleus to cytoplasm and back, doing its work in RNA, gets all localized in the cytoplasm as inclusions in the neuron. And you can see this. The brown color is staining for Tdp-43. This is a big aggregate, and you can see that the nucleus of the neuron doesn't have any inside now. It should be full. And that means that all the functions that this protein does in the nucleus are missing, and that may be the cause of the disease. ALS, amyotrophic lateral sclerosis, is not the only disease that have aggregation of proteins. Parkinson, Huntington, Alzheimer have a similar path, but with different proteins. So the frontotemporal lobar dementia are a atrophy of the frontal and temporal lobes of the brain with increasing ventricles of the brain. And the amyotrophic lateral sclerosis, if you look here, also is an atrophy in general of the brain. In particular, this is an NMR imaging of the gyrus. The problem in ALS is that the motor neurons lose activity and the muscle get wasted, so leading to paralysis and to respiratory paralysis that eventually causes death. You may have noticed that I mentioned to you two diseases that were clinically separated for a long time. Since 2006, people have realized that there are different expressions of the same problem. These inclusions of Tdp-43 that cause some problem in the neurons. Well, to show you again the inclusions and the lack of protein. So the first question is, if this is the protein that makes the inclusion, has something to do with the disease? It's just a consequence of the disease? Is of any use to follow people and know at what point I can tell, oh, you are about to develop this disease, we better take some measures. So one possibility is to use it as biomarker. Up to now, nothing good has come from here. To try to model the pathological features, for example, in animals, so we can then see if there are any therapeutic strategy. ALS and FTLD there is no therapy. ALS patients die within 2 to 5 years of diagnosis. What is the role of this protein in the neuron? Why is it so peculiar in the neuron? And obviously, are there any mutations? I am not going to talk about this and this. These, yes, there are mutations, but only 3–4% of the patients have it. 95% of the patients don't have any mutation in the protein. So the possibility is that there are two. These inclusions are toxic, and the cell dies because it's poisoned by the inclusions. Or these inclusions are just trapping the protein that was in the nucleus, don't letting it go back, and eventually causing the same that a knockout of the protein. You know, we can now, with recombinant DNA technology, remove a protein from the cell. There is one technique that is called Rna Interference, that we put a little bit of RNA there, and there are a system in the cell that degrades the messenger for that protein. And so we can test those things. We always favor the loss of function model, this one. So the first thing that we did is, let's try, if we remove the gene, as the fly is so similar to humans in this protein, obviously, we said, let's remove the gene in the Drosophila. It's very easy to do it there. And you can see here, this is the Tdp-43 gene that is called TBPH in Drosophila. And you see, this is the normal protein, and this is the flies where we have removed the gene. So there is no protein. The flies develop. So you have first the worm, then the pupa, and then the adult fly. And you see they are similar. This is the mutant, this is the normal. But this one is paralytic. And it cannot get out of the pupa by itself. You have to help it open it, the crust. No? Can show you a little bit. This is a normal fly, and this is the movement of a normal fly. This is this type of fly that die after a day. You see that it's totally uncoordinated. I cannot hold itself standing up. That one doesn't move. It's bad, but not that bad. You see that does not have coordination and not have equilibrium. However, as I told you, that fly if I put the human gene in the neurons of the fly, that in Drosophila against is easy to do. I cross it with a fly that carries the human protein as a transgene, then I recover movement. That's a transgene for human DNA, human Tdp-43 in the motor neurons only, or in the neurons in general. This is another one. So, why is that? I mean, what will happen is that, you know, as you saw when I showed the slide of amyotrophic lateral sclerosis, the neuron reaches the muscle and sort of branches out and makes a synapsis. And that synapsis in the neuromuscular junction is a series of branches with synaptic buttons. This is a normal one, you see, very ramified, with a lot of ramifications, etc. And this is the one that lacks Tdp-43. It's atrophic, doesn't have ramifications, and the buttons, the synaptic buttons that are these white balls, are completely deformed. Instead, this one is one recovered with the transgene. So we know at least one problem. Our protein is essential for neuromuscular junction formation. Now, but there is a lot of this protein in the brain, just that it's all aggregated and is non-functional. So we have all this information. Now, what can we do with all this information? I mean, essentially, here we have the bedside with the patient, and here is us doing all these nice things with the flies. So the question is crossing this bridge without ending down there. So how you get from here to something that could be useful for the therapy once you understand the disease. One possibility is, well, if I could reproduce the aggregation in an animal model and see that the symptoms are the same as the lack of Tdp-43, I can then try ways of removing it or preventing it with drugs or other means. So first thing is to do the model. And the first thing to do the model is to be able to produce the aggregates when I want. Once I can do that, I can test it in cells, I can test it in the fly, or I can test it in mice. Now, during the research that we were doing to see what the protein is doing, we found that the protein, in certain conditions, if we added this peptide that is part of the protein itself, but is a prion-like sequence, we observe in the gel things of very high molecular weight that we thought could be aggregates, precipitation of the protein. This signal is all Tdp-43. And I don't know if you can see it well, we find that if we put some sequence of Tdp-43 and many repeats of this amino acid sequence, 12 in this case, we get beautiful aggregation. This is the way that Tdp-43 staining looks like in a normal cell. These are the nuclei, what you see green, the cytoplasm is around. Can I? No. You see instead here that the signal for Tdp and for this one is red, is all in the cytoplasm. And this I obtain inducing with tetracycline this gene in the cell. This that is a construction of recombinant DNA that we do. We can put one gene in the cell that will be silent unless we put tetracycline in the media. And you can see that now the nucleus are empty. The nucleus here are blue, and all the Tdp-43 that was green is together with the red spots as aggregates in the cytoplasm. So these structures look very, very similar to what you see in the patient. And obviously, we don't know what in the patient caused this, but this is what you see in the neurons of the patient. So, here you can see it better. You see the empty nuclei. And you see here the colocalization. Let's go faster because also, when I get aggregation, I can test if the functionality of the protein is there. And this is a splicing test. The protein is important in some splicing process. And look at here, you have two forms of this messenger. When you remove the protein, if you remove it by RNA interference, you get this. And if you remove it by aggregation of the protein, you get the same thing. You get only one band. So it's working in the sense that we are depriving the cell of the Tdp-43 function. So let's go back to the fly. And we put this molecule that induced the aggregation, this bit of Tdp that induced its own aggregation in the fly. And then tested the fly for ability to walk and for the formation of aggregates. And you can see here the test. You have to look at the first 3 tubes and the difference with these 5. These are the ones that have aggregates. The only thing that the fly has different is that it's producing those multiple copies of the peptide that induce aggregation. So now you shake to pull them on the bottom, and you see this one, how they climb. I mean, and these ones remain at the bottom, and this one was up. I don't know why the video is getting stuck, but I think that the picture at the beginning was clear enough. I make you see again the beginning. So these flies has a locomotion problem, while these one don't. So do they have aggregates? And I have to finish. Yes, we can do in the larva staining of the brain of the larva, where the motor neurons are. You can see that there are aggregates, these green spots. These are the nuclei of the motor neuron. And those green spots actually sequester the Tdp protein that should be red. And you see that goes all to this yellow spot, green and red. So apparently the aggregates are working like in the human patients. We are doing the same with the mouse, and we are going to be able, by the way that we constructed the recombinant mouse, to induce only in the brain, only in the spinal cord, or in the whole mouse, according to what we wish to do. And just to end, a little bit of optimism. So we started to test ways to reduce the aggregation and recover some active protein in the cells. And we found a few substances. Here you have cells, the nuclei and the cytoplasmic aggregates. And after treating with an effector that in this case was a simply polyanion like this transsulfate, you see the reduction of over 50% of the aggregation. We are now testing some of these substances in the flies to see if we recover locomotion. Because I forgot to tell you, in the flight takes 2 weeks to be paralytic. The first 2 weeks, they are normal. And that's okay, because the flies live 60 days. So 20 days is almost 50, let's say, in human age, a little bit earlier. That is where the onset of the disease happens in man. So to summarize and finish in 47 minutes, we have the yellow dots are Tdp-43 that shuttles from nucleus to cytoplasm and helps to mature the neuromuscular junction. In the disease situation, some event makes it to aggregate in the nucleus. The nucleus tries to produce more to compensate, so these aggregates grow. When this reaches a stage that the nucleus cannot compensate, then there is no more protein in the nucleus, and its function in the neuromuscular junction ceases, and you lose the connection. So that's more or less all what I wanted to tell you. These are all the people in my lab. Thank you very much.
Thank you, Professor Baralle, for this wonderful lecture. Before we give the opportunity to the audience to ask, I suppose, sophisticated questions, let's
hope not.
Let's have a short chat and talk maybe about a little bit more general things
Sure.
that concern science and the eventual future of the field. To start with, what makes a young scientist to go and work for Nobel laureates?
Well, talking about myself, I don't know young scientists in general. I was always enthusiastic about doing research. I mean, that was what I wanted to do all the time. And the fact that I went and ended up with a Nobel laureate was a bit casual. Obviously, I applied for several places in England, including Cambridge. I obviously wanted to go to Cambridge. At the beginning, I have a series of problems, but at the end, I got in one place in the LMB, and then I moved to Sanger's division, and everything went right. In Argentina, it was the obvious choice. The Institute of Leloir, not only was he the director and he was a Nobel Prize, but was the best institute to do research within the university. So it wasn't that difficult to choose. But there's no need to be a Nobel Prize. I am not a Nobel Prize, but I like young people to come to work with me.
So it's basically it's the subject itself, it's the conditions that are given.
Mainly the subject, but you know, the subject, the condition, the luck that you have at times, no? Because I started as an organic chemist, and then moved to Leloir's lab because they needed an organic chemist. And again, in Sanger's lab, they needed an organic chemist to do oligonucleotide synthesis at the time. So it's all, I learned to do oligonucleotide synthesis, I end up studying neurodegeneration. It's not, it was not a career that was planned, essentially.
But obviously you have a good feeling for groups that do excellent research. What would be your prediction for the next Nobel Prize in chemistry or fields that are close to?
I don't know. I have been, there was one Nobel Prize that was not Nobel Prize until 2 years ago that I thought that was an injustice. It was not in chemistry, it was in medicine or biology. That was John Gurdon. He was the guy that in the 60s discovered essentially the stem cells, so the capacity of the nucleus to be reprogrammed, and then got ignored all the time. And I always thought he should get it, and he got it last year or the year before. The next one, I don't know, because it's very variable. It's very variable. In chemistry, obviously, I would like that they goes toward the side of protein structure or protein function. But they also are very alternate. No? They tend to give also to basic physical chemistry as well. And
Yeah.
so
But Nobel Prizes are always given for the research that has already been done.
yes.
What do you think is the future of
Research.
molecular,
that's what you
molecular biology, genetics? What will be the
Well,
most
the
important
future of
field?
molecular biology obviously is linked for practical and economic reasons to more applied fields like agricultural and medicine. Because the tools that we have now, although we still don't understand a lot about some of the systems like splicing, there is a need and there is the possibility of doing a lot of applied work, or at least borderline work, to develop new therapies or to develop new plants or new resistance or better crops that don't need fertilizers. And all that is within the possibilities of molecular biology, let's say. In the basic side, I am a bit worried, I must say, because there is a lot of emphasis in high throughput assays. No? I mean, you do a lot of data collection, RNA sequence, DNA sequence, microarrays, and things like that. And you get tons of information that obviously give jobs to bioinformatics. But still, you don't know of that mass of information which is real and which is not. And the validation process normally is very laborious and nobody likes to do it. So I am worried about science in that sense, that it's moving too fast, to flashy things, not covering the holes that are left behind. And eventually, all the castle may crumble. No? So my, not advice, my wish is that more scientists do one system at a time, but understand it completely, and not just global genomics or global proteomics or whatever. Because, you know, it has happened to me. Look, oh well, you look at the set of genes, 500 genes go up, 1,000 genes go down in certain conditions. And then you go to look and less than 10, 15% is true. And even if it is true, you don't know why. So I think that that's something that needs sort of a rear guard activity. I don't know if it is the same in other sciences like physics.
We'll ask our physicists.
Yeah, I know, that's what I was throwing the ball that way.
Until they give us an answer, let's hope that your wish comes through in the future. And with this, I would like to ask our visitors, our audience, to come up with questions related more to the lecture which was given by Professor Baralle. We have some...
I don't believe that was so clear, so please ask questions.
But we have experts, molecular biologists.
No, but even the biology, I wasn't
also
that
chemists. You know.
Not even a curiosity. Yes.
Physicists always ask
A
questions.
young chemist.
About the interactions inside the aggregates, what holds
inside the
aggregates, what holds the molecules together? Are these just van der Waals dispersion forces? Or?
We think that the interactions are due to between hydrophobic and hydrogen bonding between asparagine and glutamine sequences that tend to associate, you know. These are all amides. And I think that when you have many together, like in the prion, they tend to distort the conformation of the protein that they are interacting with. And some distort it more, some less, according to the composition of the sequence. All QNs are like a rigid stick, so don't have if you have something more mixed, there are terms in the structure of the protein. And then get more interaction, and you get this aggregation. Funny enough, we know that this protein interacts very nicely with another, it's called HRMPA2. And it's not there. You don't find it in the aggregates. The only thing that we found in the aggregates that was worth mentioning was a heat shock protein, HSP70. These proteins appear when there is a stress and the protein conformation is detected to be anomalous and try to mend them. So appears in the first phase of the aggregates, and then after 72 hours, there is no more. The only thing that is there is Tdp-43. That gets ubiquitinated and degraded in bits. That's all what we know of the aggregation.
Can you say anything about the sizes distribution of these aggregates? Is this, are they just one size, or do you have
there are several
big,
sizes.
small?
There is a distribution of sizes, but they tend with time to cluster in one. Obviously, we don't know what happened in the brain because, you know, what we know is autopsy material. So may have been forming for 50 years or 20 years, and we don't know exactly what we are seeing. In the cells, you get, when there is a lot, they get to clump and you get only one. Yeah.
Any other questions?
Yes?
Martina?
I wonder if these aggregates can be found in other cell types, or why are just happening in neuronal cells? What is the mechanism there that they are so different from other cells?
It's a nice question. We wonder as well. No, we don't find it. We find it only in neurons. In humans, you know, a person that has died from amyotrophic lateral sclerosis or whatever, you find inclusions in the brain, but not in the liver, not in the heart. There are some type of disease called inclusion body myositis that then the aggregates appear in the muscle and not in the neurons. But I would say in general terms, the aggregates will appear in cells that live very long, not in the skin or not in the liver that they are changing and regenerating. But the neurons, the ones that we have age 5, are more or less the ones that we have age 70. No, not, they are much less, but the ones that remain are the same. And so it's possible that the metabolism of the cell in some way slows down. These changes of conformations cannot be resolved and cannot be dissolved and start to grow. And the muscle could be the same story.
Any other question?
One more, come on.
So
then we go.
if there are no more questions, or if you are too shy, then you are all kindly invited to join the speaker and all the others for informal discussion and some drinks. So let's thank Professor Baralle again for his evening.
Thank you all. Thank you.
Thank you.